Beautiful place in town. I've not been there. Well, I've been to Jonesboro one time.
As was not
Beautiful place. Oh, you're going to film us?
Okay.
I'm just testing right now. I don't know that I'm going to do the whole thing.
How long will that take?
Wait.
I don't know. This is Geoff's camera.
Okay.
I've never used them at all, so I don't know anything.
Okay, ladies and gentlemen, thank you very much for attending Mr. Jeffrey Fisher's presentation of his master's thesis. Let me give you a quick background. I first met Geoff when I offered a course in biomechanics, and he and his lovely wife took the course together. They're both engineers, and you know, they showed a lot of interest. And by the end of the semester, they, you know, we had a good, you know, talking relationship, and he became interested in the biomedical aspect and even in the medicine aspect of things. So I asked him if he's interested in pursuing a PhD in biomechanics, and he was interested, and then things slowly grew to bigger dimensions, and we talked about orthopedics. He talked to Dr. Davies right here, and he felt that maybe he wants to become a MD and become an orthopedic surgeon. Of course, you know, we were very, you know, happy about that, and we've had students in the past that have done that, but we wanted to do an MD PhD, and then, you know, we got into his research, and, you know, we decided to do an MS MD sort of a thing. Let me tell you that the scope and size of his work is compatible to a PhD. What he's presenting today is not the whole thing, but if you put the whole thing together, it could easily be a PhD thesis. But because of his issues, he wants to, you know, have a master's and then do an MD, and that's fine. Geoff, to me, is a quintessential scientist. He's very smart, extremely talented, meticulous. I have never seen anyone as meticulous as Geoff. And it's really a pleasure to work with him. The work that he's going to show you, although we designed the experiments and this, you know, the overall project with Dr. Hardy and Geoff, all of us together, but the work is done primarily by him. And he has had to learn a number of different topics and areas, very impressive. I'll be honest with you, I'm not going to understand half of his thesis because it's the stuff that Dan does, the cell biology, gene expression. I'll be honest with you, I don't understand that stuff. I understand the mechanics part of it. But anyway, it's a very exciting work, it's very novel, it's very original. Nobody else has done this work, and I think you're going to like his thesis presentation. So without further ado, thank you very much. And
well, thanks so much, Dr. Hashimi, for the kind words. And thanks to everybody for coming out. Please feel free to help yourself to the stuff in the back if you haven't. I hope you all enjoy hearing about this research as much as I enjoyed carrying it out. And I did enjoy carrying it out. There's something very rewarding about taking a project like this from beginning to end, being involved every step of the way, learning the background, conducting the experiments, and then sitting down to analyze the data only to find meaningful results. So it's a really good feeling. And like Dr. Hashimi said, this was an interdisciplinary project. It involved work in our orthopedic biomechanics lab here in this department, as well as Dr. Hardy's molecular biology lab at the Health Sciences Center. So I organized this similar to a typical journal article. I'm not sure why some of that's cut off, but we'll improvise. So I'll just discuss each of these sections in turn. I'll try to give my engineering colleagues the relevant background on the molecular biology component, and for anyone who doesn't have an engineering background, do the same for you for the engineering component. But before I enter the molecular world, yeah, that's cut off quite a bit, but it's okay. I first want to discuss with you the intervertebral disc, which was the organ of focus for this study. So on the left is a picture of the lumbar region of the spine, which is in the lower back. And you can see these little white inserts in between the adjacent bone. Those are the intervertebral discs. They serve as the soft tissue cushions between adjacent vertebrae along the entire length of the spine. And primarily what we were concerned with in this study was the cervical intervertebral discs, which are not in the lower back but in the upper part of the spine in the neck. But the composition is essentially the same. You can see, well, you can't see because it's cut off, but there's a proximity of the intervertebral disc to the spinal cord and associated nerves. This is part of the spinal cord, and it runs down through this encapsulated canal that protects it from damage, and then running out along the sides here, along the entire length of the spine, are peripheral nerves. And the proximity of the disc to these nerves becomes relevant when we talk about disorders of the disc. All right, so if we were to take out one of these discs and look at it from the top, you'd see something like this, without these cutouts, obviously. The disc consists of 3 grossly distinct regions. Only one is not pictured here, and that's the cartilaginous end plate, which is a thin layer of hyaline cartilage that separates the disc from the adjacent vertebrae. The other 2 regions are shown here, and those are the annulus fibrosus and nucleus pulposus. The nucleus pulposus forms the center of the disc. It's a kind of gelatinous material that consists mainly of water and also a large amount of proteoglycan molecules. These are proteins, modified proteins that are synthesized by the cells in that region. And then surrounding the nucleus pulposus on every side is the annulus, and it's a series of fibrous rings called lamellae, and those are composed primarily of the protein collagen. So these notable differences in the different regions of the disc begs the question of functional differences, and indeed there are very different mechanical functions of these different regions. Essentially, the nucleus pulposus, with its large water content, helps to resist compressive loads, whereas the annulus resists tensile loads. So if we were to compress a portion of the spine that contains a disc, the nucleus acts similar to air in a tire. Under the load, it expands in all directions in response, and as a result of that expansion, it exerts tensile forces on the surrounding annulus.